Rose. Join award-winning gardeners Chris Van Cleef and Teresa Byington as they chat with rose lovers and experts from around the globe. With each episode, you'll gain valuable knowledge and insights to achieve the rose garden you've always dreamed of. Listen now as we explore the world of roses.
Hello, friends. Today I'm joined by Dr. David Byrne, professor and Basye Endowed Chair in the Rose Genetics Department at Texas A&M. Now, Dr. Byrne will walk us through the research journey of sustainable roses. Doesn't that sound good? From the early work to today, and share the wonderful news of funding for new work that is of great interest to those of us who love the rose. Hey, Dr. Byrne, welcome to Rose Chat.
Well, thank you.
It's so good. I'm just excited about our chat today. We're going to look at where we've been. We're going to look at where we're going with sustainable roses. But I want to start with your background. You actually worked with two men who are giants in the rose world.
Yes, that's how I got introduced to the rose world, actually, because I was hired here at Texas A&M in '83 as a peach breeder, which I still do, and I've released 20 or so varieties for the southern U.S. But in the early 90s, I met Dr. Basye. And for those who don't know who he was a retired mathematician out of Texas A&M and a very enthusiastic rose breeder. And now he didn't breed to release things. He just bred for fun. And his objective was to develop things that were well adapted to our hot, humid climate. And I think one of the most disappointing things I ever experienced when I started working with roses is I put out a large variety trial and I saw how poorly adapted most things were. You know, Mr. Lincoln should be great, right? Well, it dies here unless you protect it. Yeah. And so that was a sort of wake-up call for me. And when I met Dr. Basye, I basically knew nothing about roses. I wasn't thinking about ornamentals. I was into the fruit world. And he began to introduce me to this. And actually, I met him because he came by my office one day and he asked me if I could count chromosomes. And I mean, that may be a weird question for some, but for a breeder, that's pretty normal. And I said, well, yeah. And so he gave me some rose tips that he had. He said, can you count these? And I said, well, I guess so. And so I did. And from there on, I started being pulled into the rose world. And it's been quite a journey. And with him, I started learning more about rose germplasm, and he was interested in various species and getting resistance for black spot out of species and put them into the cultivated germplasm so we could use them in our gardens. And so this went on for various years. I started developing more of the research genetics aspect of the work. And then later in about 2007, and I met Ralph Moore before, because when I started working with roses, the first thing I did was to visit rose breeders, because that's the best way to learn, by seeing what other people are doing. And so I met Ralph, and we've been in contact for years and years. And he called me up, or actually his lawyer did, and he asked me, would you be interested in a donation from Ralph Moore? And I said, well, yeah, I certainly would. And for those who don't know Ralph Moore, he's the father of the miniature roses. And in his lifetime, he released, about 500 different rose varieties, of which about 300 or so were miniatures. And his emphasis was really on unique floral traits. So, of course, he had the miniature. He worked a lot early on stripes, the halo trait, mossing, cresting, various things like that. But he also was interested in sustainable roses as well. Mm-hmm. And so I had the opportunity to be able to visit with him extensively, to talk to him about his breeding, and again, learn a lot from him. So I had those two mentors that really taught me quite a bit of rose breeding. And so sustainability was sort of the beginning of it.
I read that though Dr. Basye and Ralph Moore's names are often linked, and they are, especially with this work, and they knew of each other's work, they never actually met.
No, they didn't. I was trying to get them to meet, but they were when I met them, neither of them were traveling much. So we couldn't get them together.
Well, Dr. Byrne, let's get to the basics. And could you share what we mean when we say sustainable rose?
Okay, sustainable roses. To me, that's a rose that's well adapted to your environment. One that you don't have to spray all the time, that just sort of takes care of itself. For the humid South, you talk about resistance to diseases, environmental stresses. For us, it'd be most likely heat stress, sometimes soil issues and things like that. If you go further north, you're talking about cold tolerance and things like that, in other diseases. So your disease package and your environmental resistance packages that you need might be a little different depending on where you are. And that's why you see regional trials of roses now. And I think that's an excellent thing to have. So it's something that's easy to take care of. You can grow without sprays or much management. Of course, it needs to be attractive. I mean, any commercial rose has to have all the horticultural package there as well. I did a 2012 survey of rosarians and rose growers, and I asked them the question what they want in a new variety. And the number one request was disease resistance. And then later in a later project, we did a willingness to pay study. The marketing people here at A&amp did this study. And what they found was the trait that people were more willing to pay most for were the adaptation traits. And the traits they looked at was disease resistance, drought tolerance, and heat tolerance. And that's pretty particular to our area. But if you go to other areas, you'll probably see other adaptation traits. Again, adaptation traits were the most valuable, more valuable than the horticultural traits when you're choosing varieties in this study.
Occasionally, I get the opportunity, especially in the spring, to speak to different master gardener groups and about roses. And a lot of master gardeners don't grow roses, so I'm thrilled to talk to them about the beauty and the fragrance and the wonderful things about roses. But the one thing they want to know, will I have to spray it?
Yeah, exactly. And that's one thing you're trying to get around. Again, when I first did my first variety trial and had things like, Mr. Lincoln and JFK and all these popular varieties, they really didn't do well in Texas with our disease pressure and our heat. And so it was very disappointing. It was very hard to grow a rose. But now we have a lot of other shrub roses that are much easier to grow. And so variety choice makes a big difference.
Oh, it absolutely does. So tell us a little bit about your beginning work with sustainable roses.
Well, it's in the beginning of sustainable roses. Again, Dr. Basye was talking about adaptation, and that's part of sustainability, of course. That's one of the key points. And of course, he released things like Basye's Blueberry, Basye's Purple, Belinda's Dream being an earth kind and really well adapted to our environment. So the first work went kind of slowly, and mainly because there was little funding available for ornamental research. So I looked around, and this sort of changed in about the mid 2005, 6, 7, somewhere around there. The federal government opened up a new funding program called the Specialty Crop Research Initiative research program. And this is USDA. And so that opened it up to specialty crops, which are mainly horticultural crops, because before all the other projects were open to all crops. So corn and soybeans and things like that took most of the money. And so there was very little left for us. And not that we didn't have good projects, that we just overwhelmed. And so my first proposals focused on genetics and breeding of black spot resistance. These were not funded, but I also worked with other people who worked on some of the breeding efforts to accelerate breeding. And we can talk about it a little more. And so in 2009, we actually got some funding on a project called RosBREED. And this project, what it focused on was developing molecular marker technology to accelerate our breeding projects. And it didn't work on just roses. RosBREED is a project on the rose crops in the rose family. And so the main emphasis was fruit crops. But as I was a peach breeder, I was involved in the fruit crops, and I pulled in roses as well. They didn't complain since they like roses. But we had peaches, we had almonds, we had apples, we had blackberries and strawberries all mixed in here. And so we got a little funding for roses, and I started working on molecular aspects of that and focusing again on black spot for the most part. Now, this sort of began the concept of accelerating our breeding through molecular markers. Let me sort of explain how that works and why it was sort of exciting for us. If you look at rose breeding in general, you're talking about a 5-year breeding cycle. And you may not have context for that. If you're talking about corn breeding, you can do 2 to 3 cycles a year. So roses is a lot slower. And so generally what happens, you make the cross the first year, grow the seedlings in the greenhouse the second year. And if you're selecting for something like Rose Rosette resistance, it takes you 3 years in the field to do that. So it's a 5-year cycle. Now, the idea is to use your DNA information and develop sequences that are associated with specific traits. And so those are called markers. So it's sort of like a signpost that you can find out where you are on that chromosome. And so you know which little piece of DNA is going to or where it's going and whether your plant has it. And that DNA is associated with a certain trait. And of course, my interest would be disease resistance. And so if you can do that, you can do the DNA sequencing on small plants in the greenhouse. So what follows is that instead of having a 5-year cycle, you can do selection in the greenhouse after 2 years. And so your cycle is a 2-year cycle. So it increases your rate of breeding tremendously. It doubles it, a little more than doubles. But in addition to that, since you're selecting the greenhouse, you don't have to take as much out in the field. Mm-hmm. So instead of taking, all your plants out in the field for evaluation, you'll take maybe half of them, or actually maybe probably about 10%. So you're decreasing your work in the field and the amount of field space you need for this. And then in addition, you know what your parents have, because you can, instead of just saying your parent has a certain trait, you know what genes it has. So you can make better choices on which parents to use in your crosses. So you have various advantages. Of course, that was 20 years ago when we started this work. And at that time, it would not have been practical to use the technology because it was too expensive. But we've come a long ways.
We certainly have. We hear a lot about DNA in plants today. In fact, I've noticed several of the plants that I've known as a certain name for all of my gardening years, those names are now changing because of DNA. So yeah. It is definitely changing, what we do, and that's a good thing. Now, I think it was probably in 2015 when I first heard about the research that was being done on RRD. I had found it in my garden, although through the years I found very little. I'm very lucky in that regard. But I was at a rose conference in Nashville, Tennessee, and Dr. Mark Wyndham, my first meeting of him, he introduced us to this pesky little mite and this horrible virus. I often say Mark Wyndham can make anything funny, but it was difficult for him to dull the impact of this news, but he tried. So let's talk about some of those days in the initial work.
Well, the initial work, and actually when I started working with roses, Rose Rosette was not on my radar. Again, I've been focusing on black spot. It wasn't until 2013 when the industry organized a Rose Rosette workshop. And they called me and said, hey, can you give a talk on breeding for Rose Rosette resistance? And I was sort of there, yeah, I could. I guess I better read up about Rose Rosette because I've never seen it. And this was in 2013. And so I started educating myself. And that industry organized conference was organized by Star Roses and Plants and the Rose Garden Council, which is the organization that represents a lot of the propagators. And they brought the industry, the public extension people, research people, various government agencies. We all got together and we discussed Rose Rosette. And I learned a lot. Mm. And one thing we wanted to do was to essentially develop a plan, which we did, which evolved into a proposal. And that proposal was submitted and was funded in 2014. So you have to realize at this time, we just beginning to learn about Rose Rosette. We knew it was out there, and we'd known about Rose Rosette for at least 70, 80 years, because it was first reported in the 40s and 50s. Rose Rosette is a North American native. We can't blame anybody for bringing it.
Shoot.
It's us. But its cause was only described in 2011. So we didn't know what caused it. We had suspicions, but we never proved it. And so in 2011, it was described. And so when we put this proposal together in 2013, 14, we really knew very little. So we had to do diagnostics. We didn't have a test to verify we had the disease or not, beyond by looking at symptomology, which can be confusing. We had to do a lot of field trials to just learn more about the biology of the virus and the mite, to explore control methods, how it's distributed, where it's distributed, because we only had a vague idea of that. And then, of course, from a genetic point of view, we needed to figure out what was resistant. And at this point, we had some guesses, and so we created some populations to study resistance. But a lot of what we did was to put a large variety trial in. We planted it in Texas, in Delaware, and with Mark Wyndham in Tennessee to evaluate for Rose Rosette resistance in Delaware and Tennessee. And in Texas, we looked at black spot resistance and cercospora and a lot of other horticultural traits. So we did a lot of this, and we discovered one very important thing is that all major commercial varieties are susceptible to Rose Rosette, which, was not what I was hoping. I was hoping I'd find this wonderful resistance. It didn't come. There are levels of susceptibility. So there are some that are less susceptible than others. So we learned that. We did find some species, North American species particularly, that were immune, as far as we could tell, from Rose Rosette. And so there was some immunity there, but unfortunately, those do not breed well with our commercial materials. So using them would be a long-term project. And not that breeding is short-term, but this is even longer term. So you're talking about decades. Mm-hmm. So anyway, so that's sort of what we started doing. So we're doing these field trials. We also had money to do socioeconomic analysis. Again, the willingness to pay study was one of them we did. The economic team also estimated the value of a Rose Rosette resistant variety, and they estimated the value about $35 million per year. Wow. And in reality, you can't just have one variety that's resistant, because we have 100 major varieties, right? And so we have to convert all these things to more resistant varieties for garden use in the Midwest and the East Coast anyways, where Rose Rosette is an issue. And so there's a lot of work to be done. And of course, Extension Group put together the Rose Rosette website, which is a repository for all the good information on Rose Rosette. And unfortunately, there's a lot of bad information out there too. But this is where you should go for the good information. We started working on a distribution map as well. And so asking for input from our Rose Rosette network. And so we did various things like that. And in parallel to this, the RosBREED project was refunded. And so this went on for the same time we're doing the Rose Rosette work. And in this, we put a lot more effort into the black spot resistance. And in the new iteration, we involved the University of Minnesota and University of Wisconsin. So Stan Hokeson and Dave Slezak were involved. And they worked on vertical or dominant gene resistance to black spot. And then Texas A&M would work on partial or qualitative resistance on black spot. And these are really two very different types of resistance. And so if you look at those and distinguish between the two, you have the vertical, and sometimes called dominant, sometimes called qualitative resistance. But basically, it's a sort of a lock and key type mechanism. So the varieties that are resistant have locks, and the pathogen has keys. And we talk about varieties of roses. If you look at the pathogen, it's not all the same. They're essentially races of pathogen. And each one has a different key to try to get through the resistance barrier, which is the lock, right? And so you get resistance to a dominant type resistance, and it is resistant to a set of races. But if you get a race that has that key to get in there, it's susceptible. So it's what we call race-specific resistance. And that's the resistance that Stan Hokeson at the University of Minnesota and Dave Slezak, the University of Wisconsin, River Falls are working on. They've done really great work. They've identified several vertical resistance genes, and they continue working on that. Now, the other type is partial resistance. Now, as it indicates, it doesn't give you complete resistance, whereas the other one is yes or no. This one is different levels of resistance to the pathogen. So I sort of look at it as the strength of the door to get into the plant. And the higher the resistance, the stronger that door is. But it can be overcome if there's enough pathogen on the outside to overcome it. So what this does is just sort of, it slows down the reproduction of the pathogen on the plant. So it doesn't eliminate the pathogen's ability to reproduce, but it slows it down tremendously, so you have very little disease. And so the more genes for resistance you get, the higher level of disease. And so from 2000 to 2019, during these two projects, we learned a lot about both of these pathogens and improved our ability to genetically characterize roses. As molecular techniques were improving, it's amazing. It's sort of like computer technology. Molecular technology is just accelerating the progress every year. And it's getting less expensive now, so it's becoming within the capability of breeders to use it to accelerate their breeding. But in 2019, the funding stopped. And we still had a lot to do.
So when the funding stopped, tell us what was still on your to-do list.
Oh, it was so big. For one, we still had field work to finish up, because we had started, especially the genetic work, we had started that and got that in the ground in 2018. So the funding stopped in 2019. So we still had that to finish. We still had problems with analyzing genetic information. And the problem here is that all of a sudden, a lot of the tools we had were for diploids. And let me sort of step back now. Most animals and plants are diploids, which means they have two sets of chromosomes. So you have, if you look at the probabilities of things going here and there, there's just, one or two. But most roses are tetraploid, so they have four sets. So again, mathematically, it's just more complex to analyze. So we had that. And then we had a lot more data too, because now we had molecular markers. So we didn't have hundreds of data points. We had millions of data points to analyze. So we needed better software to deal with the number of data points, but also with the complexity of the tetraploid organisms. Of course, we had to continue breeding work. We still had to find new sources of resistance because we found some. And then there's been continuous breeding, so we have to confirm some of this work as well. So we had to continue with that. We had to now understand the mechanism of resistance, especially for Rose Rosette resistance. So we found resistance that actually was derived from Rosa wichuriana. So now we had to understand why it was resistant. Was it resistant because it was affecting the virus biology, or was it affecting the mite biology because it's transmitted by a mite? So those are important with respect to going forward and learning how to use the resistance and how to manage the disease. Of course, we had to identify additional genes because we only had a few and we knew more were out there. And then once we did this, the next step is to put all those genes from a breeding point of view into one variety that would be sustainable and resistant to both black spot and Rose Rosette. So at this time, as the funding ended, we still had field plots maintained in Texas and Tennessee. And so we had to find funding. And
so tell us, where did the funding come to continue the field work?
Well, we were lucky that both Texas A&M University and the University of Tennessee helped with some funding. Of course, Robert Basye endowed a chair in rose genetics and had a little endowment funding to work with, so I used that. The American Rose Research Trust was key as well. They were very good about continuously funding the work to keep it going. The Rose Garden Council came through with some funding to get things replanted and to do the evaluations. Pat Shanley and Paul Zimmerman stepped in with a GoFundMe campaign. So we had hundreds of Rosarians donating money towards the project as well. And I thank every one of them for their donations. And again, without this funding, we would have lost several years of research. But we were lucky we had the industry behind us and the community behind us to keep this funding working. So that kept us going. And so my next step was to put proposals together again. And so we put a sustainable rose proposal together. This was in 2020. We put the first one together, I guess. And then we also, it's 2019 actually. And we also put a tools for polyploids proposal together. And this gets into my real nerdy part because it's all about genetics. And what I ran up again is what I was explaining to you previously, the diploid versus tetraploid. So again, working with roses genetically is much more complex than most plants. And thank goodness it's not just roses. Potatoes are like this. Sweet potatoes are like this. You get blackberries, you get strawberries. So there's a lot of other major crops that have this issue in what we call polyploidy, tetraploid, hexaploid. So they have more than two sets of chromosomes. And again, up to this point, if you looked at the software or the computational tools we had to analyze this data, it was mainly for diploids and for corn and soybeans and inbred crops. Well, our crops are not inbred for one, which makes them a little more complex as well. So this whole project, it brought together breeders from various crops, potatoes, sweet potatoes, blackberries, turfgrass, and rose, and then brought the computational scientists, the people who now knew how to make the codes to analyze all this. We had people from North Carolina, from University of Wisconsin, from Wageningen in the Netherlands, and from the Plant Food Research Organization, New Zealand, and actually at Texas A&M to come together to actually develop new software and improve the existing software so we can analyze this stuff properly. Because up to this point, I've been trying to use the diploid software to analyze the tetraploid. And it gives you results, but not always with a lot of confidence. And so I put this new project in, and it actually was funded in 2020. And so that was the Tools for Polyploids, especially crop research initiative. And again, it was key to being able to analyze our rose data properly. And so that's been going on we've just completed 2.5 years, and we've done tremendous work at improving the software. It's been a really exciting project.
As a rose grower, this is all getting very exciting.
Oh, it's as a breeder, it's really exciting. And so the next step was to actually get money for roses again, but just for Rose Rosette or black spot. Actually, I wanted to combine them now. Because again, the ultimate goal when I started in the 1990s was a sustainable, well-adapted rose variety, right? And so I needed both diseases in there. Because when you ask people and you look around in Texas and in the US, black spot is very important. And of course, black spot throughout the world is extremely important. And Rose Rosette in the US is the most important disease at this moment because it kills the plant. Whereas black spot defoliates, but generally doesn't kill the plant. It just makes it ugly for a while. And, I have plants in front of my house that I've been trying to get rid of. And I finally did because we had to do some renovations because they had wonderful flowers, but they had never had leaves. But my mother-in-law wouldn't let me take them out because she'd like the flowers. And I didn't like them because they didn't have leaves. But she wins always. But so anyways, we've come a long ways in developing things. So now we have better tools to analyze data. Sequencing is becoming less expensive. Technology to develop and use DNA tests to figure out which genes you have there are increasingly easy and less expensive. So the development of a sustainable rose project, which combined all this plus the extension and the economics and marketing part, we had all the pieces in place now. And so we started pushing this a lot harder and trying to get this new project funded. And then finally, in 2022, it was funded. Now, this was after the third attempt. So every attempt, you get a little better. Because you get feedback from the reviewers and they tell you, oh, no, we're not going to fund it like this. And so it's all about what they will fund. And so it's always a process. You think, okay, we submit something, we have funded, we're fine. It never goes that way. It always, in these projects, the Specialty Crop Research Initiative projects gets funded if the industry supports you. So it's a very complex procedure. We get support from industry, from the community, and you get matching as well. So industry has come through with, we requested in this last project, $4 million of funding. Mm-hmm. So we had to come through with $4 million of matching funds. Now, some of this is through my salary and salary of everybody on this project. I think we have 17 people on the project. And it includes Texas A&M University, Oklahoma State University, University of Tulsa in Oklahoma. You have the USDA in Maryland, University of Tennessee, University of Georgia, and Ohio State University. I don't think I missed anybody, but if I did, I apologize. I don't have the list in front of me. But so there's 17 or so PIs that help us with this project. Mm-hmm. So it's very complex. And I had to get like 40 letters from industry, committing donations of plants, funding to sponsor meetings, locations to do research, help with communication, which the podcast is doing that here, and a lot of different things. So it's been quite a process, but we finally got it funded in 2022, and we have the overall ultimate objective was, again, to develop sustainable rose cultivars. Again, gardeners want easy to grow, disease-resistant, sustainable roses.
Yes, we do.
And breeders want to produce them because they want to sell their roses.
Yes, they do.
And so it's a win-win for both sides. And I work sort of in between people saying, okay, well, we want this, and this is how you can do it quicker. And so that's my role here. And so we've come a long ways. And so this project, again, a big part of the project is the genetics of it. And again, we're trying to find more sources of resistance. And so we're doing trials, both of genetic populations for genetic studies as well as varieties in Texas A&M, University of Tennessee, and now we'll be at Oklahoma State University as well. So we have those 3 locations. Right now, I'm trying to get things planted. So we're getting a lot of plants in and organizing things. And so we'll do variety trials and genetic trials there. We're trying to find more genes for resistance to Rose Rosette. And that we'll be doing these trials. And we're finding sources from some of the Canadian varieties. And then, of course, we're just doing a general variety trial to see if we can find other things that might be resistant. We're looking for more sources of resistance to black spot as well. So people up in Minnesota and Wisconsin, they're looking at 3 more sources of resistance for vertical resistance to black spot. They're trying to characterize those. And they do a lot of their work in the lab. And then I take all their populations and put them in the field as well to see if they have any partial resistance to them. And so we're doing it both ways. And so both black spot and Rose Rosette is progressing. And now along with that, we're doing the molecular work to find markers for the resistance we know about so we can accelerate our breeding process and start combining these genes into one genotype or one plant. So, basically, normally what I see, I find a good resistance gene for Rose Rosette in variety A, and for black spot in variety B, and for a different black spot resistant variety C. Now I got to take those and intercross those and put all those resistance genes into variety ABC. Mm-hmm. So that's basically what we want to do. We want to do it as quickly as possible. And with the genes we have, we figure it's going to take about 3 generations to put them all in one package. Okay? And that's 3 generations and, tens of thousands of plants later. So it's a lot of stuff we have to go through to get this done. But if we did it the old-fashioned way with 5-year generation times, you're talking about 15 years. Now, if we condense that to 2-year generation time, we can do it in 6. Mm-hmm. So that's a big difference.
Oh, it is a big difference. All of this is just truly great news. And, I just feel like this project could not be in better hands. And, but what about the rose community? Tell us some ways that we could continue supporting this effort.
Okay, there's a couple ways. One way is interaction with the extension people. The extension program is developing, this year they'll be planting out a couple variety trials just for demonstration purposes and collecting data to confirm what we know and maybe learn a few more things about these varieties and how they react in different environments. Because one thing we need to be careful about as we incorporate resistance to some of these diseases into these varieties, make sure the resistance holds up in different environments. And so we're going to try to hopefully plant these in Texas, Tennessee, Georgia, and Ohio, and perhaps other places to see what the environmental influence are on these various disease resistance genes. And so that's one thing we're going to do. They're going to put other trials looking at control of the virus. They have some what I'll call antiviral substances. I don't know if that's really the proper term for them, but somehow they suppress viral growth within the plant. And so they want to put a trial out on that. They'll be working with the Rose Rosette website and improving that, adding more information about resistance. So what we've learned thus far, we'll put that on the website so it's easily accessible. We also have a Rose Rosette network where we sign up people and they report whether they see Rose Rosette in their garden, so we can get a better sense on if Rose Rosette's moving further away and to new areas, because we really don't know that. What we have now is we have reports in certain areas where extension people are, and we need to expand that to other areas to get a better feel for how well adapted it is, or how well spread it is. I don't know if well's a good term here. How widely distributed the virus is. And so we need to do that. We can also get, getting help spreading the word and good information about Rose Rosette. If people can put articles in their newsletters, talk about it at their meetings. I know Mark has talked at a lot of Rose meetings about Rose Rosette and black spot and management of both diseases. I'm willing to do the same, though sometimes I can't travel quite as much as Mark, and so I might have to do it by Zoom, but I'm more than happy to do things like that. We will have good information out that you can pass out and things like that. I'll be talking at the American Rose Society about our work as well, so if you're at the annual meeting, come by and listen to me.
You've been listening to the Rose Chat podcast with Chris Van Cleve and Teresa Byington, expert rose gardeners who want to help you achieve the rose garden of your dreams. Don't miss an episode. Listen anytime on our website at rosechatpodcast.com or listen on the go via the Rose Chat app on iTunes and Stitcher Radio. Share this podcast with your social networks and join us on Facebook, Twitter, and Instagram by using the hashtag RoseChat. Join us next time for another edition of the Rose Chat podcast. The Rose Chat podcast is a production of the Rose Chat Media Group, Birmingham, Alabama.